This is a production of Cornell University.
Larry? Yeah. All right, so my name is Zach Stansell. I am the USDA Agricultural Research Service Hemp Germplasm Curator at the Plant Genetics Resources Unit in Geneva. So, amongst some other things I'm doing as well. Anyways, so in Geneva, the USDA has a really big presence here, and we maintain crop genetic resources collections. So for example, we have the largest apple collection in the world. Y'all kind of probably saw it on McCarthy. We also keep a collection of about 6,000 tomatoes, which is amongst the largest in the world, and honestly many other crops. So in 2020, we really got started on this project of adding hemp to our germplasm collections. So why would we do this? Well, there's a lot of reasons. Maintaining crop genetic diversity, I believe, is an intrinsic good. Maintaining diverse cultivars from unique geographic regions long-term can solve a lot of problems in the future that we may not even be aware of now. For example, with cabbage, we maintain a cabbage accession that was collected in the 1890s, and it still is available. And many times these older cultivars are sources of R genes or resistance to different pathogens, pests, etc., or contain novel phenotypes. So what you're seeing right now is our first grow out of about half of our collection. So since I was here last year, we had 0 accessions. Now we've got about 200. Our plan is to grow that to 2,000 in the next 10 years. So we're really just getting started. We are trying to not just conserve this genetic diversity, but make it available to the entire hemp breeding and research community. So essentially, germplasm requesters can request a sample of the seeds from every one of these accessions and have them sent anywhere in the world, right? We are also extensively phenotyping this collection for kind of a mind-boggling number of traits. So plant architectural traits, height, width, growth architecture. Every female plant is going to have tissue sampled and sent off for cannabinoid analysis, terpene, anthocyanins, phenolics. We're taking a subsample of seed for fatty acid analysis. We're trying to chop every single plant out of this field at the end of the season and send to our fiber collaborator, Christelle Home, at the New Orleans USDA Fiber Lab. And he's going to be evaluating every plant for many different fiber characteristics. So we'll get all these huge data sets and attach them to each accession. So a plant breeder could say, like, I want a CBG dominant cultivar with low apical dominance that has low pectin, whatever you can think of. Like you can find germplasm that fits the set of criteria that you're looking for. So that's just a little bit of what we're doing. I'm going to pass it off to our hemp breeder, Tyler Gordon, as well. By the way, if you want to look in the field, you can scan the QR code on the left, and that will link you to Grin Global, which is an online page for each one of these accessions. It's kind of depauperate of data right now, but by end of season, we should have a lot more info there.
Can you hand me the other mic too?
Yep.
I'll hold that. Is that okay? Okay. All right. Thank you all for attending this afternoon. It looks like a terrific crowd out there, and we've heard some fantastic research studies that have been ongoing this morning, and so really excited, like Zach, to share a little bit more about the USDA ARS collection. My name is Tyler Gordon, and again, I'm with USDA ARS. I came just recently, a few months ago, from the USDA ARS lab in Idaho, and there I worked on small grains. So the grain part of hemp I'm very familiar with. The secondary metabolite profiles and fiber not too familiar with yet. However, as Zach mentioned, we're going to be doing lots of evaluations this year. I think we're going to take over 100 individual trait data points from each one of these plants here. And that's going to be super informative for breeders, regardless of what the end product is that you're breeding for. So whether that's fiber or grain, secondary metabolites, or maybe a combination of some of those things. I think that'll be really useful data. That'll be posted on the USDA GRIN Global website. So anyone throughout the world has access to that information. Additionally, I think Zach mentioned this, the seeds from this collection are going to be available to the research community. The one stipulation that we have from these seeds is that they, in order for us to distribute them, they're going to have to be low THC. So if you're looking for those high THC cultivars or varieties or accessions, you're not going to be able to get them from us. One thing that I would ask of you guys, so we're just kind of starting this pre-breeding program here. I'm hoping that you have access to the QR codes. I want to hand around our flyer here. And if you would, scan the QR code. There's a survey there, and I would like everybody to kind of, if you're interested in hemp material for your breeding programs, if you could scan the QR code, fill out the survey. It's very quick. I think there's 10 questions or something, and that can kind of help us determine what kind of breeding priorities we should set for this material. So we'll have all this terrific data from this location. We also have a couple other locations that we're going to be collaborating with on the West Coast, so Oregon State, Washington State. Also hoping to develop collaborations with some groups in the Midwest and also in the South to see how these plants perform across geographical locations. I think some of the other folks today have talked about the importance of some genotypes might look terrific here in Geneva, but they wouldn't necessarily look fabulous, say, in Louisiana or Alabama. And then one other thing I want to talk about just a little bit is some of the disease and insect resistance that we heard about this morning. That's, I think, one of the really amazing things about these collections is how they can act as a reservoir for important genetic diversity and also important genes that we might not even know we need for the crop at this point. And so, for instance, if there's a new disease that we're not familiar with and it comes in 10 years, that's what these germplasm collections are here for, is you might have a really ratty looking, you can see some of this material out here where it's a landrace or a ditchweed from Nebraska, and it looks like it has no value whatsoever, but you screen that against some new insect or disease and you can find some really interesting resistance there. And so again, all this data is going to be made publicly available. The germplasm is also going to be available to the hemp community. Please fill out the survey, the online survey. That's really going to help us. Oh, yeah. And just one other point, I wanted to thank our field team. Zach and I did not do all this work ourselves, and so I see some folks out here, Nick, Dan, Tony, I saw Peter earlier, and so thank you so much. And I also wanted to thank the organizers, Cornell, for letting us highlight this terrific diversity in hemp. And so if you have any questions, please let me know. Also, please feel free to walk through the material. As Zach said, where is that? Oh, you have the sign again. At the start of each plot, is one of these signs. There's a QR code that'll link you up with GRIN, which is our online, the USDA online genetic database. On the front side, it gives you some more information like the field number. The back side has the cultivar, if that's maybe more familiar with people. And so I can take questions at this point, or you can ask them at any point.
The signs are on the north end of the plot?
They're serpentine.
Serpentine.
Oh.
So you're going to have to kind of look
Figure
for
that
them.
out.
Yeah.
So let's say, right, you're sending all these samples to be tested out. You're measuring 100 different variables of the phenotype, but the expression of the phenotype depends on a lot of factors, right? So how do you average over many seasons, or there's many sites, or how does that work?
That's a really great question. So Green Global, which is, like Tyler mentioned, our online database, is really tooling up to capture these robust high-dimensional data sets. So kind of the old model was you had one trait for that the tomatoes are yellow. The new GRIN model is such that we can attach large data sets that can capture multi-year, multi-environment trials and keep those attached to not only the accession, but the inventory, the seed lot within that accession. And then also, as these collections are genotyped, those markers will also be attached to the accession as well. Great question. Come on, there's got to be a few more questions in here.
If I may.
Yes, go for
So
it.
then similar for the genotype, let's say you're preserving, you're preserving varieties, cultivars here, but if the cultivar originates from a different environment, is there a worry, or how do you deal with potentially the genotype drifting or adapting to the local environment here?
Yeah, so that's the classic question in crop genetic resource conservation, right? So there are many different factors to maintain that allelic diversity, the heterozygosity within a population. First of all, we don't even really know how heterozygous a lot of these materials are. I would guess a lot. There's a lot, but that probably varies from cultivar to cultivar. So in a perfect world, you would maintain an infinite population size and store a zillion seeds from that random mating. You can't do that with our seed bank size and capacity, and it's a few of us. So how do you find the sweet spot of optimal population size, pollination control, running regions from older seed lots? There's a lot of best practices that you can take to maintain that genetic diversity long term. You didn't ask this question, but our seed lots are also split up and stored in multiple locations in the event of something catastrophic happening. So we have a mothership gene bank at Fort Collins, Colorado, that we'll send aliquots of the seeds to. And then those seeds in turn will get backed up at the Svalbard seed vault in Norway in a quote unquote black box. So that's something that's very much on our mind, and as hemp has not been conserved in a rigorous scientific way for the most part, there's a lot we're trying to learn. So I'm pointing out some of the work of Nick Jenna, my postdoc here. So he's looking at how do you store pollen long term. There's just a lot of open standing questions that we're trying to drill into that will, provide a cohesive set of principles to maintain that genetic diversity long term.
And I just want to add really quickly, you've probably heard from some of the other groups today about the heterogeneity levels within things that are even classified as cultivars. And so pay close attention as you move through the field today. Look, you can see a label, and then there will be 10 plants behind that label, and you'll see they don't all look alike or even close to alike in some cases. Sometimes within one cultivar you'll have some plants that are 5 feet tall and some that are less than a foot tall. And so that's something that's one of the priorities for the breeding program is to release stuff that has a lot more uniformity within the cultivar.
Great, thanks very much, Tyler and Zach. We are very, very fortunate to have the hemp germplasm repository here at Cornell AgriTech and PGRU. We thank Senator Schumer for that. His office made the initial push for that location, and we really appreciate that because it's great to work with these folks, and we have immediate access to their knowledge and resources. And
It goes the other way.
Yeah.
to us as well.
Yeah.
This has been a production of Cornell University, on the web at cornell.edu.